This second part of talk is going to be really focused on genetic testing of monogenic autoinflammatory diseases. So the first gene identified in autoinflammatory disease was actually a gene that is found in patients or mutated in patients with familial Mediterranean fever gene, and this gene is known as MEFV. So here shown is what happened during the past 20 years. Basically today there are more than 30 genes that are associated with different monogenic autoinflammatory diseases. And despite this major progress in identification of all these disease-causing genes, still mutations in these genes account only for about 40% of the patients that are clinically diagnosed with autoinflammatory diseases. Most genes will be discovered in the near future with the use of next-gen sequencing technologies, with improvement of the algorithms that we use for data analysis, and also having better access to these large databases of variant allele frequencies in different populations, which are very important for gene discovery projects and also for doing genetic diagnosis of these patients. So here shown are a list of dominantly inherited monogenic autoinflammatory diseases, and basically what I would like just to say here is that these patients have single or heterozygous mutations in respective genes, and these mutations are passed from one to another generation. Patients that have very severe phenotype, as is the case, for example, with patients that have SAVI or NOMID disease, they have what we call de novo mutations. So these de novo mutations are not found in parents. Here is shown a list of recessively inherited monogenic autoinflammatory diseases, and these patients have two mutations in a given gene. We call them B-allelic mutations. Typically, these mutations are loss of function or hypomorphic mutations for most of these diseases, except for the gene or mutations that are associated with familial Mediterranean fever. They are recessively inherited, but they are considered to be a gain-of-function mutations. There are more genes linked to autoinflammatory phenotype in the context of broader immune dysregulation, and this is the concept that has been increasingly recognized during the past few years. So in the past, we tend to group the patients into patients with autoimmunity or autoinflammation or primary immunodeficiency. So what we are talking more about nowadays is that there is this spectrum of immune dysregulation in patients that have immune diseases, so that in addition to autoimmunity, patients may present with some features of autoinflammation, or there are patients, for example, that have primary immunodeficiency. They are susceptible to various infections, as is the case with Lubach deficiency or RIP kinase deficiency. However, they may also have component of autoinflammation, so they present, for example, with GI inflammation. So what is the purpose of genetic testing for systemic autoinflammatory diseases? Is to confirm a specific diagnosis in an individual patient or a family, and to exclude all other conditions that present with overlapping phenotypes. So this genetic testing is not a simple process, for the reason that inheritance of these autoinflammatory diseases is not straightforward. So Hal already mentioned dominantly recessively inherited diseases. However, in addition to this type of inheritance, there is also evidence for digenic inheritance in patients that have proteasome-associated autoinflammatory syndromes, also known as CANDLES. So these patients have two mutations, but instead of having two mutations in one gene, they have two mutations in two different genes. And typically, these genes encode the proteins that make up or compose a large multi-protein complex. So most patients that have PRAS or CANDLE have B-allelic mutations, two mutations in the PSMB8 gene. However, what you can see here is that there are some patients that may have one mutation in the PSMB8 gene and the other mutation in another gene that encodes for the protein that also makes up this immunoproteasome complex. So basically, essentially, it means if you have a patient with suspected CANDLE disease, we can't just screen for mutations in one gene. At this point, we are screening for mutations in 6 genes, but it's also indicated to screen for mutations for all the 14 genes that are involved in the making up of this complex, or that make up this complex. There is also evidence for oligogenic type of inheritance, and this is basically a type of inheritance which involves co-inheritance of several what we call low-frequency variants. So what are these low-frequency variants? So these are variants that are found at the allele frequency between 1 to 5% in general population. So patients that have rare monogenic disease diseases, they have either novel or very rare variants. Patients that have this oligogenic type of inheritance, they have basically several of these low-frequency mutations, and then patients that have what we call complex or polygenic diseases, they basically have co-inheritance of several common allele variants that are found at a frequency of 30 or 40% in general population. And for these patients, such as patients with RA or lupus or Behçet's disease, basically we say that they have presence of several what we call the susceptibility allele to mutations, but not real true pathogenic variants, because they are found in unaffected people as well. So, because these variants are low-frequency variants, they are often found in unaffected parents of these patients, and typically these patients present with non-specific and milder immunoinflammatory phenotypes compared to patients who carry novel or rare variants in the same gene. So the hypothesis here is that presence of several of these low penetrance or low frequency variants basically may have some sort of synergistic effect on some inflammatory pathway to induce inflammation in these patients. However, that has not been demonstrated by any functional studies. And examples of such variants that are very common in our field are R92-2Q mutation, P4612 mutation in TNF receptor 1 gene, or E148, 369 mutation in the MEFE gene, or several variants that are identified in the NLRP3 gene. So when we find variants, these low-frequency variants, basically these are what we call non-confirmatory genotypes. So, they don't confirm diagnosis of any particular autoinflammatory disease. So Hal already discussed patients with somatic mutations, and basically typically patients with autoinflammatory diseases have these somatic mutations in the myeloid cells, and these somatic mutations can explain or account to up to 30% of the patients that are diagnosed with cryopyrin-associated periodic syndrome, or CAPS. There are also reports of somatic mutations in patients with SAVI, with NLRC4-associated MAS, and also hypoinsufficiency of A20. Hal also mentioned that there is evidence for gonadal mosaicism. What does it mean? It means that you have two, for example, affected siblings. You look at the parents and they don't have symptoms, or they have very mild symptoms, but basically, the fact that there are two affected siblings in this family suggests that there is a gonadal mosaicism in this family or in one of the parents. And to prove presence of somatic mutations basically requires NGS-based testing technologies, either of a single gene or targeted gene panels, or VES or whole genome sequencing. We can test for the presence of mutations in peripheral blood mononuclear cells or myeloid cells. However, we cannot really test for the gonadal mosaicism. So why do we do genetics testing for systemic autoinflammatory diseases? There is a lot of overlap in clinical manifestations among these patients. However, and in addition, there is also significant phenotypic heterogeneity. By that I mean that mutations in one gene can lead to very different phenotypes, as I will illustrate in the following slides. So neutrophilic dermatosis, for example, or skin inflammation, is described in patients with different autoinflammatory diseases. And here you can appreciate this pyoderma gangrenosum type of skin rash, which is seen in two patients, and clinical presentation is very similar. Nevertheless, these patients have mutations in two different genes. So this patient basically has a mutation in the MEFE gene and has a disease which is known as PAAND, while the second patient has a mutation in the PST-PIP1 gene, and nevertheless they present with very similar skin inflammation. Interestingly, these two proteins, PST-PIP1 and pyrin protein, which is encoded by the MEFE gene, are interacting proteins, and basically they are involved in regulation of the same inflammatory pathway, namely IL-1 beta signaling pathway. Here shown are also patients that are diagnosed with another rare inflammatory disease, and that is pride. And again, these patients, in addition to other features, I'm not downplaying the role of clinical manifestations, which are important for the clinical diagnosis, but nevertheless, they also present with the features, severe features of skin inflammations. And finally, here is shown patient that present with severe skin ulcers, and this patient turned out to have a mutation in the TNF-AIP3 gene, which regulates totally different pathway, NF-kappa B signaling pathway. So another examples are, for example, patients that present with early onset monogenic vasculitis of small vessels. So these two diseases are known as SAVI and DADA2. these patients basically present sometimes with very similar clinical features as shown here, peripheral skin ulcerations, olivedoid type of skin rash. Nevertheless, they have mutations in two different genes that are inherited in different ways. So patients that have SAVI, they have gain-of-function mutation in the STING protein, and which drives basically interferon signaling pathway, while patients with ADA2 have recessively inherited loss-of-function mutation in the ADA2 gene and protein. Further evidence for genetic heterogeneity, patients with cold-induced autoinflammatory disease. The first disease described with this phenotype was basically FCAS1, or mutations in the NLRP3 gene. Today there are at least 5, and many more genes will be published also soon that are linked to cold-induced autoinflammatory disease. Most patients have mutations in the NLRP3 protein. Nevertheless, we have to now consider screening for mutations all these other genes. So here shown is another family, two siblings that were provisionally diagnosed with early onset lupus, monogenic type of lupus, because they presented with specific skin rashes, such as this discoid skin rash, which is seen on the cheeks of this patient, and other type of skin rashes. So nevertheless, one of these two siblings also had or presented with strokes, and that was the reason why the clinicians considered possible diagnosis of DADA2. And basically, these two siblings were proven to have DADA2 or deficiency of ADA2, both by protein functional testing and also molecular diagnosis. So again, autoimmune phenotype is described in the spectrum of DADA2-associated phenotype. Nevertheless, it could be misleading. I mean, clinical manifestation in these patients were more suggestive of monogenic lupus than of DADA2. So there is also examples of mutations in the same gene that can cause two very different phenotypes, and that's certainly the case with mutations in the PLC gamma 2 gene, which are linked to PLAD and APLAD. So patients that have PLAD have heterozygous deletion, which are shown here in this gene, while patients that have APLAD, which is quite different, clinically quite different phenotype, have single base substitutions or missense mutations in the same gene. Both types of these mutations, deletion and missense substitutions, affect the autoinhibitory domain of this protein and basically act as a gain of protein function mutation in this pathway, and they upregulate the activity of many downstream molecules. So there are also examples that mutations in the same gene could be inherited in different ways, or that they may have different inheritance pattern. And I will illustrate that on the example of pyrene-associated diseases. So most of you know of familial Mediterranean fever, or FMF, and basically this disease is linked to recessive mutations in the C-terminal domain of this protein, B30.2 domain of this protein, and most patients have two mutations in this domain. There are some examples of patients that have one mutation, and we believe that these patients probably have susceptibility allele to inflammation in other genes. So there are some rare examples of patients that have only one mutation in this protein domain. However, there are patients that have clearly dominantly inherited mutations in other domains of this protein, and these patients have disease which are known as PAAND and dominantly inherited severe autoinflammatory symptom that used to be called FMF-like, but it's really nothing like FMF because these patients have much, much more severe phenotypes. So what is the explanation for that? So basically, these FMF-associated mutations are sort of we call them gain-of-function mutations, but essentially they are hypomorphic mutations because they affect the function of this autoinhibitory domain, B30.2 domain, and they basically just reduce the threshold for the activation of this protein, while these other mutations that are inherited as a dominant trait, they affect the residues which are basically leading to mutations, and these residues lead to the constitutive activation of this protein. So in the case of PAND patients, these mutations affect these two residues, which are important for the phosphorylation of protein. So there are three types of clinical genetic testing that we can discuss today: single gene testing, multi-disease genome panel testing, and clinical exome that Hal also mentioned. This clinical exome basically involves reporting only clinically significant variants and copy number variations, but essentially is done by whole exome sequencing or whole genome sequencing. I would also like to mention today a new paper that is put together by the international team of geneticists who are experts for the genetics of autoinflammatory diseases, which basically proposed best practice guidelines for the genetic diagnosis of these 8 autoinflammatory diseases: FMF, mevalonate kinase deficiency, TRAPS, CAPS, BLAU, PAPA, DADA2, and hyperinsufficiency of A20. So what is shown in this paper, or what we proposed in this paper, is that if the patient presents with a clinical phenotype which is highly suggestive of specific SAD, as is the case, for example, with patients that present with fevers and they come from at-risk population, as is the case with patients with FMF who come from the Mediterranean population, or if there is a positive functional test that are done in patients that are suspected to have DADA2 or mevalonate kinase deficiency, then we suggest that these patients should basically test it by single gene, Sanger basically sequencing for mutations either in specific exon or all the exons of a given gene. So if this single gene sequencing is not informative, then the next step to do is for patients that have, for example, DADA2 and mevalonate kinase deficiency and hyperinsufficiency 20 is to search for copy number variations, because there are reports of patients that basically have copy number variations as the disease-causing mutations. In patients that present with CAPS-like, BLAU, or TRAPS-like disease, if the single gene sequence is negative or we don't find confirmatory genotype, then the next step would be to search for mosaic mutations, because there are reports of multiple patients who carry mosaic mutations in myeloid cells leading to respective phenotypes. So if this search is again non-confirmatory, the next thing would be to run targeted gene panels for these 8 genes or more genes if available, and if this testing is negative, then the next step would be to do whole exome sequencing or whole genome sequencing. Patients that present with non-specific phenotype should be tested either by targeted gene panel sequencing or should be tested by VES. So here you may wonder, why do we go through all these steps, instead of going straight and doing targeted gene panel sequencing or whole exome or whole genome sequencing? And the advantage of doing single gene sequencing is just that it's faster. For example, we can do results in a couple of days if necessary. We can have the genetic testing result reported. And it's also less expensive than doing whole exome sequencing or whole genome sequencing, although this technology has been increasingly now used at NIH and many other institutes and hospitals. So here I would like to give you an example of what we had to do in one family to basically prove that this family has a DADA2. So here shown is the family from India with unusual inheritance pattern. So basically there are 2 affected siblings in this family. The father is also affected, so you think of dominantly inherited disease. However, there is consanguinity in this family, which would then suggest that they are more likely to have biallelic mutations in some gene. This family was provisionally diagnosed with Damon-Blackman anemia, and they were sequenced for mutations in a number of DBA-associated genes. No variants were identified. Because some patients with DADA2 may present also with DBA-like clinical manifestations, they were suspected maybe to have DADA2. So we did Sanger sequencing of ADA2, and we didn't identify any pathogenic variants. Testing for DADA2 could be also done by functional or protein test, and this is the next step that we did, and basically show that these 2 affected siblings and affected father had absence ADA2 activity. So this is a confirmatory test. This is a protein test which is unfortunately not available for many autoinflammatory diseases, but in case of DADA2 it is available. So why do we need to go a step further? Why do we need to do now sequencing of this gene? So basically, the importance of that is really for genetic counseling in this family. So what we did initially was to screen by Sanger sequencing for missense changes in the coding region of this gene. However, copy number variations are also reported as the cause of disease, DADA2. So for that reason, we developed another assay in our lab, which is called MLPA assay. And by running this assay, we showed that both affected siblings and the affected father basically were homozygous for duplication of exon 7 in this gene. So you can see that we had to do multiple levels of testing in order to basically find what is the cause of the disease in this family. So targeted gene panel sequencing, Hal already talked about them. So they're time effective. Typically, you can get your results in a couple of weeks. Relative to whole genome or whole exome sequencing, they're cost effective. They cost less money than doing whole exome or whole genome sequencing. They have higher accuracy than VES or VGS because they have higher depth coverage, and that's why they are more indicated to find somatic mutations, copy number variations. The other advantage of doing targeted gene panel sequencing for for geneticists is that basically there is lower likelihood to identify variants of unknown clinical significance or VUSs that are very hard for us to interpret. They are hard for us, they are hard for clinicians, they are also challenging for patients to understand what is the clinical significance of these variants, right? So the recommended depth of coverage for clinical sequences is at least 30x over single nucleotide. And so the next question is, now we have all these beautiful technologies and access to public databases, something that we couldn't even dream of 20 years ago or 30 years ago when I started doing genetic studies. So what is the diagnostic yield and clinical utility of targeted gene panel sequencing? Unfortunately, for most diseases, with exception for some intellectual disabilities and some neurodevelopmental diseases, it's not higher than 30%. And that is certainly the situation with monogenic autoinflammatory diseases. So what are the possible explanations? Why we don't have why there is no better diagnostic yield? So we need better algorithms for in silico analysis. We need population-specific allele frequency databases. This is something very important because all these public databases are based on the frequency of these variants, mostly in Caucasian populations. So we are still missing these population databases in other, ancestries or other populations. So there is this issue of reduced penetrance, which is complicating interpretation of these results. There are also cell-specific somatic mutations that may not be detected in peripheral blood samples. I mean, for them, we have to really sort out the cells and to do sequencing in myeloid cells, for example. And this technology also does not capture non-coding variants and does not capture large copy number variations. By that, I mean large deletions like 100 megabases or something like that. They are not going to be detected by this targeted gene panel sequencing. So here I would just show you a couple of studies that reported what is the yield of targeted sequencing for autoinflammatory diseases. So this study was done in the UK, and basically they evaluated 55 patients with undiagnosed autoinflammation and vasculitis for mutations in over 100 genes, and their diagnostic yield was only 10%. So only 6 out of 50 patients had at least one clearly pathogenic variant, class 5 variant, how we call it. 3 out of 6 were definite cases. The other 3 patients were presumably diagnosed, but actually not completely. They didn't have confirmatory genotype because 2 of these patients had monoallelic pathogenic variant in this gene, which is associated with HLH. In order to confirm diagnosis of HLH, which was not their clinical presentation to begin with, one would really have to find 2 mutations in this gene, and another patient was a female carrier for X-linked Wiskott-Aldrich syndrome mutation. So they also claimed that 22% of patients in this cohort had at least one likely pathogenic variant. So the overall yield was no higher than 30%. Diagnostic yield was not higher than 30%. Here is an example of another study which nothing to do with autoinflammation, but these are the patients with primary immunodeficiency tested for mutations in 571 genes, and conclusive diagnosis was made only in 60% of these patients. Another good outcome of this study was that they identified these 3 patients that presented with atypical features, and basically they confirmed their diagnosis by identifying mutations in respective genes. So how to interpret the genetic testing? Well, that's the bottleneck. So we have to do a lot of filtering for low coverage variants, anonymous variants. We ignore these anonymous variants, although theoretically they could be also disease-causing variants. We also filter out variants that have allele frequency higher than 1%. I prefer to do filtering even with a lower number. I filter variants that have frequency higher than 1 in 1,000 because they are less likely to be pathogenic. Fortunately, we had access to these large databases that are shown here. And as I mentioned, these population ancestry-specific databases of variants are also very important. If they're not available, the next step is always to do testing of the parents. We don't test always trios. We don't test always parents. Typically, we test probants, but parents are very important, especially for patients that come from specific ancestries. We also have access to this InFevers database of gene variants that are identified in patients with systemic autoinflammatory diseases, and this database, which is very useful for all us geneticists, basically has been recently curated by expert geneticists for mutations in 8 genes associated with autoinflammatory diseases. So if you go now to this InFevers database, there is a column there which will clearly say whether this mutation is pathogenic, likely pathogenic, or benign. And if in doubt, it's always good to do segregation analysis in a family. And we also rely on use of these in silico prediction tools, which are shown here, POLYF, CAD, SIFT, and so forth. However, we have to be sort of reserved when relying on these in silico prediction tools for the reason, for example, that FMF-associated mutations that have been known now for a number of years and shown in thousands of patients are predicted to be benign. On the other hand, you can see a variant that has very high CAD score, but they are very common in the general population. So they are helpful, but they are not absolute. So here is shown the standardized DNA variant scoring system that was developed for monogenic or Mendelian human diseases, which is also used in patients with monogenic autoinflammatory diseases. So this is what you're going to see on the report from diagnostic companies. It's either pathogenic, likely pathogenic, variant of uncertain significance, likely benign, or benign. So the most complicated, of course, are these variant of unknown or uncertain clinical significance. So what does it take to make confirmation of clinical diagnosis of a patient with suspected monogenic autoinflammatory disease? So confirmation of a dominant disease requires presence of a pathogenic or likely pathogenic variant, class 4, class 5 variant. Confirmation of recessive disease requires 2 pathogenic or likely pathogenic variants, and they should be shown to be inherited in trans, which means they had to be shown to be inherited from 2 different parents. Combination in trans of a pathogenic or likely pathogenic variant with a rare or novel VUS should be reported as consistent, not confirmatory, but as consistent with the clinical diagnosis, because we are not sure whether this novel VUS is really functionally relevant variant. Identification of a de novo variant may further support its genotype pathogenicity. A common VUS is not regarded as a confirmatory genotype, and basically this paper recommends that only novel rare VUS and class 4 and class 5 variants should be reported. Predictive diagnosis. Well, for diseases that have a risk of irreversible damage, such as, for example, NOMID or DADA2, predictive testing of asymptomatic family members may be justified. This is something that should be discussed with the family, whether they are ready to do it or not, whether they want to know the result or not. So follow-up of these at-risk individuals may avoid occurrence of life-threatening complications. Whether such asymptomatic cases should be given prophylactic treatment remains controversial, and that's the case, for example, with DADA2. So our practice here at NIH is to treat these asymptomatic patients that have molecularly confirmed DADA2 with TNF inhibitors to prevent occurrence of complications of disease. But that is not the case in many other hospitals and institutes in the US and also in other countries, because this treatment is very expensive. So in summary, genetic testing is now an important part of rheumatology, allergy, immunology practice. Clinical diagnosis is hampered by lack of clinical criteria and functional tests for most of these diseases. Genetic testing is considered the most valuable, if not essential. I don't want to overestimate the importance of genetic testing, because obviously that's what I do. I do molecular diagnostic testing, and I don't want to underestimate the importance of clinical diagnosis, but it is really, becoming very, very important for diagnostic purposes. And genetic testing is considered not only in differential diagnosis of SAD, but can also assist clinicians in the process of choosing an appropriate treatment. So if you get your result and you see mutation in NLRP3, you know that this patient should be treated with IL-1 inhibitors. If you get, a result that says that this patient has a mutation in TMEM173, you know to go to and use JAK inhibitors, etc. Advances in genetic testing technology have increased detection of pathogenic variants, and we have certainly seen that during the past 20 years, but also complexity of data to analyze. And we cannot ignore the role of environmental and epigenetic influences and factors that can modulate the clinical phenotype and that should be taken into account for genetic counseling of patients and families. So going back to these two cases that were presented earlier in this talk. So basically the result of targeted gene panel sequencing of case 1 was not very informative. So basically this patient was found to have 6 variants of unknown significance, including R92-Q and Q703-K mutation in TNFR1 gene and NLRP3 gene. So what does he have? What do you do with a patient like this? So I mean, obviously one can think of doing VES or VGS, which would be even more complicated for analysis and more expensive, etc. But if this patient presents only with fever, what I would do personally, I would just follow up this patient for a little while to see, whether he may develop some other more specific, manifestations, clinical manifestations, and that can help also a lot, even if you get his VES data with data analysis. So we can focus on certain pathways, maybe, and not to look at the entire exome data. The second patient was what we say solved case. So this patient basically was shown to have a somatic mutation in the NLRP3 gene, in the exon 3 of this NLRP3 gene, and this mutation was found in 10% of blood cells that were tested. So I think I went a little bit over the time that was allocated for this talk, and I apologize to the next speaker. I don't think we have time to take any questions, but you're more than welcome to come to me, and if you want to talk about genetic diagnosis or if you have some interesting patients or whatever, I would be most happy to talk to you during the lunchtime or later. Thank you for your attention.